One more minute. This is just syncing and then we'll go live. All right, now I'm going to broadcast. Welcome, everybody. We'll be beginning in just a few moments.
Good evening and welcome to tonight's Friday evening lecture. I'm Nipam Patel, Director of the Marine Biological Laboratory. The MBL is dedicated to fundamental biological discoveries that inform human health and the health of our planet. Every summer since its founding in 1888, the MBL has hosted public evening lectures where renowned scientists, historians, and architects of modern biology have presented stunning scientific discoveries for exploration and discussion. Despite the hardships of the war years and the Great Depression, the Friday evening lectures have always been held without interruption. Just as our predecessors did, we are coming together as a community to adapt this year's series with streamed lectures delivered by notable Friday evening lecture alumni. While I regret that we can't be together in person in the Lilly Auditorium, your presence here tonight is helping us to keep this important tradition alive. Thank you for joining us and enjoy the presentation. And remember to turn off your cell phones.
Good evening. My name is Carrie Alberton, and I'm a Hibbett Early Career Fellow here at the Marine Biological Laboratory. I'm delighted and honored to introduce Margaret McFall-Ngai, tonight's speaker for the Porter Friday Evening Lecture at the Marine Biological Laboratory. The annual Porter Lecture is held in honor of Dr. Keith Roberts Porter, a former director of the MBL, considered by many to be the father of the field of cell biology. This lecture is sponsored by the Keith R. Porter Endowment, whose goal is to support communication and education in cell biology. It's an honor to introduce Margaret, who is currently the director of the Pacific Biosciences Research Center at the University of Hawaii at Manoa and a Howard Hughes Medical Institute professor. Margaret's work has been to study the interactions between animals and microbes. She has been studying the microbiome long before it became a buzzword. To study these interactions, she developed a model system in an unlikely place, the light organ in the Hawaiian bobtail squid, which hosts a peculiar bacterium that produces light. The interaction is specific to only these two organisms, and studying the communication between the squid and this species of bacteria has provided insights into the importance of microbes in every aspect of animal life, from embryonic development and gene expression to circadian rhythms. These ideas have been transformative as we begin to appreciate the importance of beneficial bacteria in our world, and Margaret has been at the forefront of this, from her work establishing the National Microbiome Initiative and as chair of the Canadian Institute for Advanced Research program on humans and their microbiome, to her Guggenheim Fellowship integrating macrobiology and microbiology. Dr. McFall-Ngai was a Moore Scholar at the California Institute of Technology, and she is currently a member of the American Academy of Microbiology, the American Academy of Arts and Sciences, and the National Academy of Sciences. Margaret has fostered a whole community investigating the squid-bacterium relationship, with more than a dozen of her former trainees now heading up their own labs studying this system. This little Hawaiian bobtail squid has also found its way to the MBL as part of our Model Organisms initiative. And on a personal note, her pioneering work has become an important part of my research program. And so with that, I'm happy to yield this virtual version of the Lilly Auditorium stage to Margaret McFall-Ngai for her lecture on the complex molecular dialogue of host and symbiont through their lifelong partnership.
Many thanks, Carrie, for that kind introduction, and thank you for the tremendous opportunity to participate in this lecture series. The MBL is a very special mecca for the community of biologists, and I will forever be grateful for the opportunity to participate in the Friday evening lecture series, as well as the summer course program. I've been involved with the summer course program for about 25 years, giving lectures in microbial diversity, developmental biology, as well as physiology courses. I'd like to extend here a special thank you to the director, Nipam Patel, and others in the leadership at MBL and at the University of Chicago for bringing the community together for these events and the invitation to speak here. It's not quite the same, of course, as being there at the MBL, but nonetheless, it is MBL summer fun. So what I'd like to do today is I'd like to talk to you about my research program. And what I'm going to start, though, with is I'm going to start with a little discussion of the context. And so this first slide shows you where I work. This is the Kewalo Marine Lab of the University of Hawaii at Manoa. And as you can see, it's right on the water there in Point Panic in downtown Honolulu. My talk will consist, like I said, of a significant portion of time talking about the context of the work, and then I will talk to you about a model system that I work on, this small little casual guy down in the lower right-hand corner. And it's a symbiotic association with a marine luminous bacterium. So let's get started. So we're going to start with what was the technology that enabled the field that I work in. So the technology that I'm going to be talking about is driving a huge change in the field of biology. So what is this technology? Well, actually what it is fast and cheap gene sequencing or DNA sequencing. So what I'm showing here is I'm showing the cost of sequencing a megabase of DNA in dollars on the y-axis against time on the x-axis. And you can see that there's an inflection, and so that the curve for cost falls off of Moore's law. In other words, the price is halved every couple of years at about 2008. 2 years later, that blue arrow was when I gave my first lecture here. And so between that arrow of where sequencing became cheap and also fast, it's gone down, and such that today it's somewhere in the pennies. So it went down from $10,000 a megabase in 2001 to pennies. So this was incredibly enabling to the community. And so how did this change biology? So imagine this. From the 3rd century BC to the late 20th century, biologists organized the biological world based on what they could see. And that is what they could see either with the naked eye in Aristotle's time to what they could see with very complex microscopes. So with the unaided eye or with the aided eye. But the problem with that was that the physical and physiological features of the microbial world or of microbes aren't reliable characters of their relatedness to one another. And so while we were able to construe a set of relationships between animals and plants based on what we could see, the features of microbes didn't lend themselves to that. And so they had no evolutionary biology. We couldn't tell their relatedness. But now we can look at genes, which is the real readout of our relatedness. So what we have here in this image is we show that the biological world, based on what we could see, was divided in the through the end of the 20th century into what we call 5 kingdoms. And this was based on something that Whitaker had put together, and this was from a publication in 1979, and it was the 5 kingdom model. And you can see that at the crown groups are things that you can really see, like animals, plants, and fungi. And then down below are the very small things that are nearly featureless, and we thought there were very few of them. Well, when we could sequence the genes, what you see on the right-hand side, you see a huge explosion of diversity. And everything that's in the fungi, animals, and plants, if you follow that red arrow, becomes that little bit of that now big tree, and everything else is microbial. So the first thing is that microbiology found evolution. In other words, they could, by using gene sequence, find out the relatedness of microorganisms. The second thing we learned was that the vast majority of the biosphere is microbial, the vast majority. And so what I want to do here is I want to step back and I want to compare it to physics. So there was a book that was written by Werner Heisenberg, one of the major players in physics in the 20th century. It was an autobiography. And in the introduction to his book called Physics and Philosophy, the person who wrote the introduction, Paul Davis, said, "True revolutions in science involve more than spectacular discoveries and rapid advances in understanding. They also change the concepts on which the subject is based. Such fundamental transformation took place in physics during the first 30 years of this 20th century, culminating in what has been called the golden age of physics. As a result, the physicist's world view has been radically and irreversibly altered." So these are the characters that created the golden age of physics. It was the 20th century revolution in physics that Max Planck, Werner Heisenberg, and Albert Einstein brought about. What about biology? Well, there have been many revolutions in biology, and the major ones, I would say, are Charles Darwin, and what we have here is in the 19th century, Charles Darwin, and then in the 20th century with Rosalind Franklin, James Watson, and Francis Crick. What they did was they took the theory of evolution that Charles Darwin had, and they found that DNA was the basis of the genetic material that was the underpinning to that evolutionary force. So the 21st century brought on what I consider the golden age of biology, and that was enabled at the very beginning, by a pioneer named Carl Woese. And he made a synthesis of these two things. And the synthesis, what he did was he used a conserved marker gene to compare that with that marker gene across all organisms and say, how are they related? And so what Carl Woese did was he made a big synthesis of both of those two things. So basically what you're seeing with this pioneering work that Carl Woese did, what you're seeing is a fundamental transformation is taking place in biology in the 21st century. As a result, just as in physics, the biologist's world is being radically and irreversibly altered. So what we found through this is that microbes are absolutely everywhere. They are the basis for health everywhere from human health. We can now define the microbes that associate with humans and what they're doing and what they do in a baby versus what they do in an adult. And so we can do that now because of what Carl Woese brought on board. And we now know that that microbes are responsible for the health of the soil, the health of the water, the health of the atmosphere. But the problem is these associations are often very complex. So how do biologists, in their daily work of trying to understand this, how do they deal with this complexity? Well, one of the things they do is they turn to animal models, simple systems that allow them to study complex characters. So on this image, I'm showing three examples of extremely complex systems in which biologists have used and are using animal models. One on the left-hand side is the going from a fertilized egg to a baby in developmental biology. That's really complex. In the middle, you have 14 synapses in the brain. Try to figure out how they're connected and how they work, how the brain works. And the last one is the 10 to the 14th bacterial cells that associate with the human body. That's a really complex system. The next thing I'm showing here is I'm showing, to give you an example of how successful models can be, I'm showing the Nobel Prizes awarded in developmental biology and physiology and medicine. On the left-hand side, I'm showing all of the recipients of every single Nobel Prize that's been given in developmental biology over the years from 1935 to 2012. And every single one of them was using an animal model. And that's how they got to the bottom of how a particular thing works in developmental biology. So what we're going to be talking about here is the power of animal models to understand complex characters like the 10 to the 14th bacteria that associate with humans. So what sorts of models are biologists studying? What sorts of things have they looked to? What experiments, essentially, has nature done with symbiosis through evolution that give us insight into how symbiotic associations evolve and persist generation to generation? So what I'm showing here is I'm showing an array of some of the models that are available, and so they can be divided into natural models that have a natural simplicity, and then there are engineered models up in the upper right-hand side. In the engineered models, all vertebrates have really complex, from fish to mammals, all have really complex microbiota. And so what they do is they take them into the laboratory, they render them germ-free, give them vitamins and all the things that the microbes would give them. But what they do is they'll put back one or a few so that they can see what microbes do to those systems. But a lot of biologists are relying on models, that some of which were used for developmental biology, but that have been used or are being exploited to understand how symbiosis works. And some of these are really simple. Some animals, for example, the honeybee, have simple consortia. The honeybee has 8 microbes in its gut, reproducibly individual to individual, that you can study and understand how a consortium is put together. That it goes down to the very greatest simplicity, which there are some invertebrates that have binary systems in which you only have one host and one microbe. So you really know who's doing what in the conversation. So what I've got down in the lower right-hand corner is the squid light organ association, which I'm going to spend most of my time talking about. So this is the squid system. So this is Euprymna scolopes, the Hawaiian bobtail squid that I've spent my career working on. And this is a system that has one host and 2 symbiotic systems. So the system that I've worked on for my career is the light organ system, which provides the animal with camouflage during the night. It's a night-active predator that goes out and hunts at night, and it uses the light produced by the bacteria in camouflage. The other organ that the animal has, all the females of this species have something called the accessory nidamental gland. I'm not going to bother you with that name, but the accessory nidamental gland, or ANG, is only found in the females, and it's a gland that has a simple consortium of bacteria in it, and it allows the animal to put bacteria on the surface of the eggs that protects the egg from fungal infection. And that's being studied by Spencer Nyholm, who's a professor at the University of Connecticut. So one of the things, because of this great change in the cost of sequencing and it becoming fast and cheap, is we were able to derive the squid genome. And it turns out that it's really complex, and we published a paper in 2009 in PNAS, and it's really large. It turns out that the genome of this little tiny squid is about 1.6 times the size of the human genome. So what have we learned with the genome sequence? Is that, and one of the most remarkable things is that we could look at these two organs and we could look at the genes that they express and ask how they might have evolved. What we found in doing that was that the light organ expresses genes that it borrowed from the eye. So many of the genes that are expressed in the light organ are ones that are also expressed in the eye. And they're very similar. Both of them handle light. One in the case is environmental light in the case of the eye, and in the case of the light organ, it's, in case of the light organ, it is the bacterial light that is handled. And you'll see in a minute how similar these organs are. But we call that evolutionary tinkering, so it borrowed from the eye. The female-specific organ is actually an orphan organ. In other words, when you looked at the genes expressed, there were no matches in the extensive databases, or many of the expressed genes had no matches, and they're what is called orphan genes. In other words, they arose specifically through rapid evolution of other previous genes to become something that runs that particular thing that is only in this squid and its close relatives. So, what I will focus on now for the rest of my talk is this light organ that I've been working on for my career. What does it do for the animal? As I mentioned, it's a night active predator, and what this animal does is it buries in the sand during the day, and then at night it comes out to forage in the water column and hunt. And so what it does is it hangs up in the water column and it waits for a prey item to go by. But it's vulnerable when it's hanging up in the water column, and so it emits light from its ventral surface of the same quality as downwelling moonlight and starlight, so it doesn't cast a shadow against the visual field of a predator looking up from below. So this cartoon gives you an idea of what counter-illumination looks like. And so the visual field of a predator looking up from below there'll be some confusion, and it will not recognize the squid as a squid and use it as a prey item. Up in the upper left-hand corner, I'm showing one of our study sites where we collect them. And you just collect them in knee-deep water and can bring them into the lab, and they're great lab animals. So let's get into the squid light organ system. So this is Euprymnoscolopes. And this casual guy, I say, if you open Euprymnoscolopes, in the lower left, you'll see that a ventral dissection shows this beautiful bilobed light organ. And that cartoon in the middle is a single lobe of the light organ, and that light organ has a lens to bend the light of the bacteria out. And the bacteria are in the red, yellow, and green and brown regions there that you see in the center of the light organ. And they produce the light, and it's bent out into the environment by the lens. There's a reflector behind them, in other words, dorsal on the animal, and that keeps the light going downward. And then the ink sac behind that acts like a choroid. In other words, it keeps stray light from going out the backside of the animal. And also the diverticula act like an iris, and they let more or less light out. So the animal effectively counter-illuminates. Suppose the moon goes behind a cloud, you want to put out the right amount of light. So if you look at a histological section, you see that the bacteria are extracellular. And so in the upper one, that's a sort of low magnification shot. But if you look down below at the TEM, where I've false colored the bacteria, you'll see that Vibrio fischeri, the luminous bacterial symbiont, is along the apical surfaces of a very complex epithelium. And so it communicates with those cells across that epithelium. So I've done the work on this system for the last 30 plus years in close collaboration with Ned Ruby, also at the University of Hawaii. And Ned and I have had this adventure. He works mainly on the bacterial side, and I work mainly on the animal side. And our whole focus is the study of the chronic colonization of animal epithelia by gram-negative bacteria. And that is perhaps the most common kind of symbiosis that is out there. In other words, that's what mammals have, that's what humans have, is the chronic colonization of animal epithelia by gram-negative bacteria. Okay, so what I wanted to do here is, with this movie, I want to show you the structure. So let me go back one and show you that this you're going to be looking at one lobe, like this diagram in the center. So what you're going to see is the Vibrio fischeri are green, and the animal tissue is magenta, and the host nuclei are blue. So this will give you an idea of the relationship of the bacteria to the animal tissue. They're really packed in there. They're really, really packed in there. And there are about 10 to the 10th bacteria. And you can see that they occur in very thin sheets in there, and are in most of the bacterial cells are in very intimate contact with the host tissues. So I should say that I'm going to have some of my students and postdocs and their names up in the upper in the pictures, so that you'll have an idea of who's actually done this work that we've been working on for many years. So I'm going to start by just giving a little information on how does it get started? How does this symbiosis get started? And so what we want to know is each generation, this animal has what is called a horizontally transmitted symbiosis. So what that means is that each generation, this animal picks up its symbionts from the environment. So just like humans, when we're born, we're pretty well sterile, but we pick up our symbionts from our mothers and from the environment. So each generation, this happens. So the question is, in the case of humans, as well as in this little guy in the sea, how is the symbiont collected? How does it select, against the background of other microbes? So in the case of the Squid-Vibrio system, there are, in normal seawater, there are 1 million bacteria per mil of seawater. So that's a lot of bacteria. And in that mill, there are only about 100 to 1,000 Vibrio fischeri. So they only make up a small percentage of the bacteria in the water. And the animal has to figure out, and the bacteria have to figure out who their partner is. So what embryogenesis does is it produces this interesting bilobed structure. In other words, on each lateral face of the light organ are 2 ciliated surfaces that that are developed during embryogenesis, and they promote harvesting of the symbiont. When the animal hatches, it begins to draw water into its body cavity and collect the symbionts along these surfaces. At the base of these little appendages that come off the ciliated surfaces are 3 pores. And those 3 pores are where the bacteria enter host tissues. And so there are 3 pores that enter, and they enter into 3 what we call crypts on each side. So in this movie, what I'd like to do is this is the ciliated field that you're looking at. And in collaboration with colleagues at Caltech and USC, we did a study in which we looked at how those ciliated fields work biomechanically to capture the bacteria. So we asked first the question, does this field of cilia size select? So I've got a little circle up there, and I want you to pay attention to what's going on. And we introduced 2 and 4 micron particles around that into the water and allowed the animal to select what it wanted. So what you can see here is that by and large, there's preference given to 2 micron particles, which are the size of Vibrio fischeri. And the 2 micron particles, they're in a region, they accumulate in a region just above the pores, and then they go in. Okay, but the important thing is most everything most of the bacteria are around 2 microns. So what else is going on? So there's a biochemical process, and what happens in this biochemical process is that they select the correct bacterium. So, but now let's ask, do all strains of Vibrio fischeri do the same thing? In other words, the species Vibrio fischeri will have different strains, and those different strains can behave very differently. So to give you an example too, that humans have several hundred species of bacteria and several thousands of strains in those species. And so what are those different strains doing? And this model can help because we can ask the squid to select a strain or whatnot. So how does that look? So what I'm showing here is I'm showing some of the strain variation that we've seen. And here, in this instance, it's strain variation in recruitment behavior. So you'll see in the left-hand image of a light or confocal image of a light organ, you'll see the green is Vibrio fischeri, and they're very tiny little aggregates of cells from one particular strain. And in the other strain, on the right-hand side, that particular strain, when you put the same number in the water column, the animal selects them very quickly and very efficiently. And they make big aggregates that go in and they actually colonize more quickly. The reason they don't sweep is because the bacteria on the right-hand side seem to do better when they're in the free-living state, whereas the ones on the left-hand side do better when they're in the symbiotic state. So there's like a balance of strategies. So now we hope to understand, using this system, the significance of strain variation in symbiotic systems. And what I'm showing you here is what this change in technology has done for the Vibrio fischeri. So the symbiont, the genome, there have been many genomes, many strains that have been sequenced. And what I'm showing here is or some of them, 62 you can find in NCBI database. But in the last 5 years, 58 of those 62 have been sequenced. And so there are a lot of sequences out there. Now, the symbiont genome is about 4.2 megabases, and it's about 1,000 times fewer base pairs than the host. So the small genome. So to recap then, this ciliated surface that you saw selects the bacteria or focuses the symbiont into regions above the areas of colonization. Vibrio fischeri gets there and then it sticks to the cilia and it communicates to the animal that they are present. And what the animal does in response is 2 things. One is it produces a chemical environment that favors Vibrio fischeri. So I was telling you about that biochemistry. It produces a chemical environment that favors Vibrio fischeri, and secondly, it produces chemicals from the pores that attract Vibrio fischeri into the pores. So it makes it a selective region, and it tells the bacteria where to go. Then what they do is they move into host tissues. So what you're seeing here is you're seeing the symbionts are aggregating on the surface, and you see mucus is the blue, Vibrio fischeri is the red, and then the cilia are green. And what they do is they move in, they chemotax to the chemicals created that attract them. Then what happens? Well, on the left-hand side here, I've left the surface so that you can see the outside portion. On the right-hand side, you see the inside of the animal. So what the bacteria have to do when they go into those pores is they have to negotiate 6 distinct biochemical and biophysical environments over the space of about only 100 microns, which are about 50 body lengths of Vibrio fischeri. So they go through a duct into a bottleneck, that sort of U-shaped area. Then there's a bottleneck just before the crypts, the blind-ended crypts, that they enter. On average, a single bacterial cell, despite a lot of them might get into the pores and down the ducts, a single Vibrio fischeri will get into the crypt spaces and grow out and populate the crypts. So let's just take a look at this bottleneck and what it does. So symbionts, when they get in there, they get, like I said, a single one gets in and then it grows out. And what that outgrowth does is it causes that bottleneck to close down so that it keeps Vibrio fischeri inside the crypt space. So if you look at the bottom image, confocal image, you'll see how Vibrio fischeri is restricted to that. It's the green that's in the crypt space for most of the day by that close down. Also, nobody else is going to get in there. Every day at dawn, Vibrio, the host animal, vents 95% of its symbionts into the surrounding seawater, and then the bottleneck opens up to allow the bacteria to go out, and then within an hour or so afterwards, it closes back down, and Vibrio fischeri grows, that it was remaining behind, grows up to fill the crypt space. So I'm going to give you one last story on the Squid-Vibrio system. And that's about, once they're in the light organ, how do the symbionts communicate with the host? What do they say to the host and how do they say it? And what I'm going to focus on here is, we've learned a lot about that, but what I'm going to focus on here is their influence. To ask the question is their influence restricted to the tissues with which they directly interact? And if so, is the response the same in the other tissues, or is it different in other tissues? And what drives that difference? Okay, so let's get into that story. Now, the backdrop of that is that with humans, for instance, we know that colonization with the microbiome influences other tissues of the host. So what I'm showing here in the center of this diagram are the microbiota. And remember, there are like 10 to the 14th cells of hundreds of species that are possible in that region. And what they do is they shed molecules that get taken up by the bloodstream and diverted to the different tissues, and those tissues respond. And so we now know that the microbiota and a dysbiotic microbiota can cause problems with the brain, with brain development, as well as it can cause things like depression and even things like autism. So there, in the human microbiome has a cascading effect around the body of a human. But how does that work? How is that done? What microbe or microbes is responsible or are responsible for a given response? And so we're asking then, with the Squid-Vibrio system, where there's one host and one microbe, do the symbionts affect gene expression in remote tissues? So what you're seeing here is a little diagram of the squid and the tissues we use to ask this question. So the organs chosen for analysis were 3. The light organ, of course, because we wanted to know how colonization of the light organ by its symbionts affects symbiosis. So you can look at non-symbiotic animals and symbiotic animals and see how they change. And then we chose the eyes to ask the same question. And the reason is because, as I told you before, there's strong convergence in the eye and the light organ. And we wanted to know how much of that is influenced by colonization with symbionts. And then finally, the gills. Turns out that in cephalopods and squid and their relatives, gills are an immune organ. And so we wanted to know how they responded to the presence of bacteria systemically. So the first thing in the analysis that Sylvia found was that if you look at the total transcriptome of both adults and juveniles that are symbiotic and non-symbiotic, what you'll find that the organs of the adults, and actually this is just a comparison with symbiotic animals, is that the organs of the adults are different from the organs of the juveniles. And so you can see that the light organ is different, the stage specific in the eye, and the gill is, the adults and the juveniles are much closer. So gene expression, this plot shows that gene expression profiles cluster with organ type, and that in each tissue type, the transcripts cluster with the life stage, and the juveniles seem to show the most variation. So if you did a total transcriptome analysis, now looking at this, I know many of you who do this for a living will say, wow, that's a lot of genes, 45,000 transcripts. There's this, like I said, there's a huge genome, and this was a very stringent transcriptional analysis. And the reason there are so many is because there's a lot of gene editing and so on and so forth. So this is a stringent analysis. But you can see that the gills and the light organ share a lot in the upper left-hand corner, that 9,796 genes. They share a lot of transcripts, whereas the eyes and the gills don't share so many, and the light organ and the eyes don't share so many as far as the total transcripts. So the gills and the eyes, the gills and the light organ share quite a few, probably because they're both largely epithelial. So, but by 24 hours, to answer the question of do remote tissues respond, remote tissues have responded to colonization of the light organ by Vibrio fischeri. And what's so cool here is you'll see that, surprisingly, the eye, although it has fewer shared transcripts overall with the light organ, it had a more pronounced transcriptomic response than the gills. And each tissue has a signature response to the light organ symbiosis. And the top 10 biological processes or go terms of each reveals a unique pattern. So the point is that every tissue studied here responds differently to the colonization of the light organ by Vibrio fischeri. We wanted to know if eyes also respond to colonization of the gut by bacteria using germ-free mouse. And so we did a collaboration with Federico Rey at University of Wisconsin. And it turns out that, that mouse eyes do respond, but a lot fewer genes robustly respond. Only 5 seem to be upregulated. 5 genes are regulated. And none of those genes are shared with the genes that are regulated in the squid eye. So we want to ask then, what is it about the symbiosis that affects the transcriptome, or the transcription in the eye, the transcription of genes? So what is it? So because they're similar to the eye, because the light organ is similar to the eye, we decided that what might be happening is that it is responding to light, and so the light production by Vibrio fischeri. So the light production by Vibrio fischeri is controlled by the Lux operon. And what would happen to the transcriptional regulation in the light organ, the eye and the gills, if you were to make a mutant in Vibrio fischeri's light production? So what you do is you make a clean deletion of the Lux operon, and that you have, with that change, the bacteria do not make light once they get into the light organ. So what effect did this have? Well, the first big surprise we had was that if you look at this pie diagram, what you're doing is you're comparing the dark mutant against the non-symbiotic animal or aposymbiotic animal, and against the symbiotic animal against the aposymbiotic animal. And what you see in the overlap is the response to bacteria. And so there's 636 genes that are responsive to bacteria, and there are, in contrast, 1,283 genes are responding to the light alone. So the greatest transcriptional response to symbiosis is the light and not to the presence of the bacteria themselves, which was a huge surprise to us. Now, if you compared the light organ transcriptomes of wild type versus the delta Lux mutants, most of the genes that are regulated, in other words, a large proportion of the 143 genes differentially upregulated, are eye and vision related, which was really amazing because it is a big response to the presence of light production. So now you ask the question about the eye. How does the eye respond to the fact that the dark mutant is not making light? So what we did was, in a 24-hour wild-caught colonized animals, 44 genes, so you're colonizing that with a Vibrio fischeri that's bright, 44 genes are regulated in the eye. And it turns out that all of those regulated in the eye are downregulated. And there are all kinds of things, oxidative stress and response, lots of them are, of that sort. So what happens with a dark mutant? With a dark mutant, you colonize the light organ. The mutants colonize beautifully at 24 hours, but they make no change in gene expression in the eye. In other words, the entire change in gene expression in the eye is due to the light production by the bacteria in the light organ, which was another huge surprise. What's interesting too is that the gills were identical in both wild type and mutant colonized. And that was probably because the gills are an immune organ and they're responding principally to the presence of bacteria. So the animal here is counter-illuminating. And so it needs to have light production. So this turns out for the eye to be a really important thing. The eye is communicating with the light organ how much light is coming down from above so that the light organ can put out the appropriate amount of light to camouflage. So light organ, the symbiont light production seems to be a principal driver of this association. And not only from gene expression, but also in development. If the bacteria are not making light, although they colonize well at 24 hours, they get kicked out of the light organ. They start getting kicked out by about 48 hours, and then eventually dark mutants are not maintained in the light organ. They are sanctioned. But also, they don't drive development. A dark mutant doesn't drive development properly, and the animal doesn't behave properly. So it's a really important factor. Now, in other animals, there are going to be other characters that drive the symbiosis. In us, it's certainly nutrition. So in summary, what I would like to tell you, or what I would hope that you would take home today with you, is that the revolution and unification of biology represents the biggest change in our view of the biosphere since Darwin. And we have some very big challenges in the field. We have to question our long-held premises. We have to, everything that we think about. At this point, we have to change education. I mean, one of the things that we know is that a biologist has been able to leave with a biology major with having a single lecture in their freshman year on the microbial world. And that can't be anymore, because we now know that the microbial world is the basis of health of the biosphere. So we have to remodel education for that's a really important thing. We have to question our premises, everything, look back and think about the way we've thought about things, and it might, change a lot of the ways we think. We need to integrate this new knowledge into our pre-existing information, and we have the goal of developing this tremendously expansive horizon. One powerful approach, I hope the second thing you bring home with you is that one very powerful approach that biologists use to understand conserved characters across the animal kingdom and to figure out how humans who are so complex work is they use model systems. They take advantage of the experiments that nature has done through evolution. I hope I've convinced you too how incredibly complex the establishment and maintenance of such a simple system like the Squid-Vibrio system is, with just one host and one microbe, incredibly complex to create that symbiotic system. So the field has a tremendously exciting future, and there's a vast arena of symbiosis out there for people to study. And this is a very exciting day. So with that, I would like to just show the MBL community a picture that we took at our 30th annual Squid-Vibrio meeting, which we had at Scripps Oceanography because I started collecting the animals, Euprymna, when I was a postdoc there. These are the academic children, grandchildren, great-grandchildren. Many of them are in faculty positions across the country, and they and their students came together to celebrate the 30th anniversary of the study of the Squid-Vibrio system. And then, of course, I'd like to acknowledge all of the funding agencies who have so generously contributed to our research. And so with that, and this little guy is settling out in the morning, coming down onto the sand and settling out to be in the sand during the day. And I'll end right there with this little movie. Mahalo nui loa. Thank you very much.
Thank you so much, Margaret, for that fantastic talk and sharing with us this incredibly exciting and powerful system in such an unlikely place. We've got a whole bunch of questions that have started coming in, but to the members of the audience, please submit them through the Q&A feature that you can see on the screen. So let's dive right in. The first question we got was, what benefit do the bacteria get from emitting the light? And is it being associated with the squid that benefits the bacteria?
That's a great question. And the why luminous bacteria make light is not entirely known, except they're facultative anaerobes. In other words, they can live with oxygen or without oxygen. And one of the lore, a bit of lore, is that it helps them move from effectively at a biochemical level from anaerobic respiration to aerobic respiration. So, you know, that's one idea. We know that that you only find Vibrio fischeri in Hawaii in areas where the squid is present. And so the squid vents 95% of its bacteria into the surrounding seawater every day. So if you have a large population of hosts, you'll have a large population of Vibrio fischeri. And so at least for the Hawaiian Vibrio fischeri, the light organ is probably their principal habitat. They are free living and they're just waiting for a baby to come by that they can colonize. So, you know, being in the light organ, I often joke with the microbiologists that the bacteria are enslaved. And they don't like that so much. They say that any environment where the bacteria can grow and with nutrients provided by the host is a pretty good spot to be. So
that's fantastic. And so one of the other questions that came up with is what is the doubling time of Vibrio, and how does the host organ provide nutrients for the bacteria's growth and sense how the population is doing?
That's a really interesting question. The doubling time in culture can be 20 to 30 minutes. And so it's a very fast growing bacteria. What happens after they vent every day is they grow back very, very quickly, such that by noon, they vent at dawn. And by noon, they're pretty much back with the population at filling the light organ. And then they go into a growth stasis, which is very interesting and poorly understood in something we're working on right now in Ned's lab particularly, is focusing on what it means to be metabolically active, but in growth stasis. So, you know, they're very quickly growing, but then they come to a halt. And this happens every single day. What they feed on early on looks to be something, probably amino acids. Your Graf years ago, while he was in Ned's lab, described that probably they were feeding on amino acids. They undergo a tremendously profound maturation. So at about 3 weeks to 4 weeks, they become fully nocturnal. You know, they do this burying in the sand and coming out during the day. At the same time, they change their metabolic activity fairly dramatically. So what happens is during the day, they seem to be using membranes provided by the bacteria to grow up right after dawn. The animal sheds membranes into the crypt space that the bacteria grow on, and that's by essentially anaerobic respiration. Then what happens is as they go into growth stasis, the animal begins to traffic hemocytes into the region of the crypt spaces, and the hemocytes deliver chitin, and chitin metabolism, which is aerobic fermentation, happens. And what that does is it drives the pH of the environment down, and the animal becomes, I mean, I'm going to be teleological and anthropomorphic here, but the organ begins to look like it's a pathogenesis. And then at dawn, with the dawn light cue, the animal vents 95% and the whole thing starts again. But that profound nutritional rhythm starts at about 3 to 4 weeks. And what's really cool is that the hemocytes are trafficking to the crypt space, and the animal has a protein called the macrophage migration inhibitory factor. And that protein is really high during the day, and it keeps the hemocytes from going into the crypts. And then at night, the animal downregulates that protein, and the hemocytes can go into the crypts. So there's this really complex dialogue between host and symbiont.
It's amazing that it's this ostensibly simple system that just has layers and layers and layers that make it actually work.
Well, it's really fun because, you know, one of the things you can do with a simple system is you'll find something like we were the first people to find a circadian rhythm in a bacterial symbiosis. And I would go to the meetings with the human microbiome people. I'd say, you know what, you got to look at circadian rhythm, and they have. And in humans, the circadian rhythm looks to be, at least in part, driven by the microbiome. And so it's really a profound circadian rhythm. So it's yeah. And so that's one of the conserved features.
That's incredible. And so outside of the circadian rhythm that seems to have, you know, a very set pattern, can the host actually control the luminosity of the light organ?
Yeah, so the animal seems to be able to control the light output by two mechanisms, by the diverticula of the ink sac. So it develops as a outpocketing of the hindgut ink sac complex during embryogenesis. And so the ink sac is sort of part of the light organ. And diverticula of the ink sac open or close like the iris in your eye to allow more or less light to go out. The other thing is that the lux operon is an oxygen requiring. It's an oxygen requiring. All luminescent reactions are oxidative. And so what happens is the animal can withhold more or less oxygen. So one of the things that happens at night when they need more light is, you know, that pH that was going down. The other thing that happens is hemocyanin traffics into the crypt spaces. And remember, in a classic Bohr effect, hemocyanin or blood pigments will release their oxygen. And so as the pH goes down, the hemocyanin releases more and more oxygen that promotes the upregulation of luminescence in the light organ when the animal needs it.
So there are many different levels to just even that functional part of its ecology.
Oh, yeah.
So another question that was raised is, do quorum sensing mutants have the same effect on the dark mutants in the regulation of gene expression in the squid eyes? Have you guys looked at that?
Yeah. So actually, I had a student back in 2008, Carlene Chun, who did a 6-way microarray in which she looked at symbiotic and non-symbiotic, but she looked at the delta lux, and then she looked at lux minus lux autoinducer mutants. And then she had a condition in which she had autoinducer mutants in there, and she added back autoinducer that they would complement. And then she gave them autoinducer alone to see what the autoinducer alone would do to gene expression. It was a very interesting thing. The autoinducer affects differently than light production itself. And there are certain things that they share in common, like the delta lux in the lux eye. The cells of the juvenile crypt don't swell like they usually do with wild type, which is an interesting phenotype. But the lux eye minus mutants, when you add back autoinducer, you can complement the gene expression. Now, what you would expect from autoinducer alone is that you would expect only those genes that are complemented to be expressed by the autoinducer alone, right? I mean, that's kind of logical. But in fact, it's completely different. In other words, well, autoinducer alone it's not the genes that are complemented by autoinducer that are turned on. So what that says is it's really common for the animal to need the presence of bacteria to properly respond to acute. And so what we found with that is that cell surface molecules of the bacteria, particularly lipopolysaccharide or endotoxin, and the cell wall components are important for cueing the host animal that the bacteria are present, and now they should respond to things like light production and autoinduction.
Wild. And so this is kind of a broader scale question. Why do the squid vent their symbionts every day?
Oh, that's, you know what? I'm not an ecologist, and I always get in trouble when I give lectures to ecologists because the bacteria do slowly grow. I mean, there's some suggestion that it discourages cheaters. Cheaters, in other words, dark mutant. The other thing is that the animals, like I said, vent 95% of the bacteria in the surrounding seawater. And if you take a baby and you expose it to water that's collected from water that's collected from where there are large populations of squid, that water is very infective to that baby. The farther you get away from adult populations, the more difficult it is for that baby to be colonized by Vibrio. Vibrio fischeri levels go down, 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 and then it's hard to get colonized. And the juveniles show significant habitat loyalty. So that is to say, they hang out with even though there's no parental care or anything like that, they sort of hang out where the parents are. So like I said, this gets me in trouble, but one side effect of venting is to seed the water for the juveniles.
And so is there a window of time that they have to be exposed to this, or are they receptive to Vibrio, whether or not they've been colonized? So if you take like an uncolonized individual and expose them really late in life, can they still pick it up?
That's a very good question. So Eric Koch was in my lab. He's now in a postdoc down at University of Florida, but he was working in my lab and he was raising the animals, and he could show that if you raise them up beyond a month, all the way to a month as an aposymbiotic animal, and what that means is aposymbiotic means exposed to all the other bacteria in the surrounding seawater, but fischeri is not present. They would always colonize. And so on the other hand, the other thing that he showed, which was fascinating, was if you colonize them persistently for 7 to 10 days, and then you cure them, you can't colonize them again. But if you cure them before 7 to 10 days and you colonize them again, they will colonize. So there's like a switch that's flipped after they've been colonized for a certain amount of time. They say, okay, I've got my symbiont, I can change, myself into a symbiotic animal. So there's some sort of learning that has gone on in that first week. But if you keep them aposymbiotic, they're colonizable at least until a month later. Now, they also looked at 4 months later, but never pulled the experiment out to see if they would colonize till then, as far as I can remember. But at 4 months, what happens is you get an atrophy of the crypt space, that crypt epithelium is not quite the same. And so a really great question is, can that crypt epithelium be resurrected if the animal's been persistently aposymbiotic? And we don't know that, yeah.
Wild. And so about, we hear a lot about the overuse of antibiotics.
Yeah.
Has this changed the microbiome of the squid in the wild? Are there Vibrio that are developing antibiotic resistance or changing due to either this, or a couple times people have asked about climate change in this context as well? Is there a change that you guys are seeing in response to that?
Yeah, it's interesting. I don't think that we know the answers to that. In other words, I don't know, because we haven't been able to, we look at antibiotic pressure. Nobody's ever looked to see what antibiotic pressure does to the symbiosis. With regard to climate change, what I can say is I can say that Vibrio fischeri will grow to 28 degrees, And then beyond that, it doesn't grow, as I understand it. And it can be alive for a few degrees above that, but it won't grow in culture. It does in the light organ. It will persist in the light organ. So the animal acts kind of like a refuge for a while there. But I suspect, as it gets warmer and warmer, there might be some effects on where the squid will maintain its symbiotic association. So what I mean by that is this particular squid is found everywhere from knee-deep water to 300 or 100 meters. And so in the summer, when it gets warm in Hawaii, it's often hard to collect the squid. And we feel that they are very likely to be moving offshore into some of the deeper, cooler water environments.
So it sounds like there's some buffer in there.
Yeah, we think at least for this there might be some buffer. We just might not see them near shore too much anymore.
Well, there's so many more questions that are listed here, but I feel like we're probably running a bit over time. I'll ask one more. So one of the questions was, does RNA editing create very different proteins in the light organs when bacteria are present versus absent? Have you looked at RNA editing in this relationship?
Oh, I would love to do that with Josh Rosenthal. So, there are so many cool questions with regard to that. Another really cool one with regard to is to what extent epigenetics plays a role. They're just, the fact is with the Squid-Vibrio system, you can study everywhere from ecology and evolution all the way down to molecular mechanism. And so the questions are kind of endless. And I know that the MBL is working on CRISPR-Cas methods. And we're really hoping that CRISPR-Cas is something that it will truly transform our ability to work on the Squid-Vibrio system. It's very exciting work that MBL is doing. And when that happens, the Squid-Vibrio system will be like rhizobium, the rhizobium leguminous plant symbiosis, and one host and one microbe with all genetics and all partners, which will be truly transformational. But at this point, that work is ongoing.
Well, this is certainly an exciting time to be asking these sorts of questions. Thank you so much for sharing this story with us, and thank you to everyone for coming to tonight's lecture. I'm sure we'll try to get through some of the questions as we go. But yeah, thank you so much, and we look forward to seeing you next year at the Friday evening lectures.
Yeah, thank you for being such a great host, and I'd like to say thanks to all the technicians and various people who made this experience possible.